Hello friends, welcome to Kns Biochemistry. Today in this video session we are going to see about one of the important area of bioinformatics, where we are going to see about the significance of the bioinformatics, where the databases are going to play a very important role. So now we are going to see about the biological databases. These databases are the resources for doing any kind of work on bioinformatics. So let us start this session. Come along with me.
The completion of the Human Genome Project lays a foundation for systematically studying the human genome from evolutionary history to precision medicine against diseases. With the explosive growth of biological data, there is an increasing number of biological databases that have been developed in aid of human-related research. In this lecture, we will present a brief overview of the major human-related biological databases, how the data is being arranged and classified into different categories according to their data types, and how to retrieve and analyze them. As human-related databases continue to grow not only in count, but also in volume, challenges are ahead in big data storage, processing, exchange and curation. What is a database? A database is a collection of information that is organized so that it can be easily accessed, managed and updated. Databases are composed of computer hardware and software for data management. The chief objective of the development of a database is to organize data in a set of structured records to enable easy retrieval of information. Although data retrieval is the main purpose of all databases, biological databases often have a higher level of requirement, known as knowledge discovery, which refers to the identification of connections between pieces of information that were not known when the information was first entered. For example, databases containing raw sequence information can perform extra computational tasks to identify sequence homology or conserved motifs. These features facilitate the discovery of new biological insights from raw data.
Biological databases, what are they? Before entering into biological databases, we must know what is a database. In the field of computer science, everyone knows that a database is a very important data, and that is going to play a very important role in computer science. In bioinformatics, what is the role of these biological databases? Databases are usually very essential for bioinformatics research and applications. So we have many databases that exist, that is covering various information types. For example, you take DNA and protein sequences. DNA is a nucleic acid, and protein, of course, it is another biomolecule. So take both DNA and protein sequence. Or you take molecular structures of any molecule, or phenotypes and biodiversity. So in all these areas, database is required. So these databases, they may contain sometimes empirical data. That is, empirical data is data which is going to give the structure or the elemental or atomic level. So they are obtained directly from the scientific experiments. Or sometimes you may get a data that is a predicted one. It's called a predicted data. That is, from the experimental data, we are going to analyze that result, and from that we are going to come to a prediction or a conclusion. That is called predicted data. Or most commonly we will have both. That is, both this experimental data and predicted data. So they may be specific to a particular organism or particular metabolic pathway, or they may be going to say about some molecule of interest. So they can incorporate data that is compiled from multiple other databases. Sometimes that the data obtained from one database is not enough. You have to get the related data from other multiple databases. So these databases, they vary in their format and access mechanism, then whether they are public or not, that is also mentioned in the database. So based on these experiments, we see that we have types of databases. First one is called primary database. So what is this primary data? Primary databases, they hold the important information that has been obtained as a result of the experiment, scientific experiment. That is, they are scientifically obtained through experiments. So experimentally determined some protein sequences, they are inferred from the conceptual translation of the nucleotide sequences, and they are called primary database. Of course, it is not experimentally derived information in case of a nucleotide sequence, but it has arisen as a result of interpretation of the nucleotide sequence information, and consequently it must be treated as potentially containing misinterpreted information. And if you go to protein database, there is a number of primary protein sequence databases, and each database requires some specific consideration. So when you say primary databases, see, these primary databases are populated with experimentally derived data that is taken from the scientific experiment. For example, nucleotide sequence, protein sequence, or macromolecular structure, etc., etc. These experimental results are submitted directly into the database by the corresponding researchers. So that is why it is called primary database. And this data are experimentally arrived, that is taken from the nature. Once given a database, and that database should be given a proper accessor number. This data which is there in the primary database are never changed, because it will be given a permanent access number. They form part of the scientific record. So it is a permanent scientific record which is given as a primary database with a particular accessor number. Now let us see about secondary database. What is this secondary database? Secondary databases, they comprise data that is actually derived from the results of the analysis of the primary data. So what we have seen, the previous one is primary data. From that result, we are going to analyze something, we are going to come with some conclusion, and that is called secondary data. They are often referred to as curated databases, but this is a bit of a misnomer, because primary databases are also curated to ensure that the data in them is consistent and very, very accurate. So now we see that the secondary databases, they are very often draw upon information from the numerous sources, that is primary sources. Even these sources may be from other databases where we have primary and secondary both. So now let us see some of the examples for the databases. For example, the database used for biological sequence analysis, example GenBank, UniProt. Then the databases which is used in structure analysis, example Protein Data Bank, PDB. Then databases used in finding protein families and motif structure, that is motif means protein structure, motif finding, Interpro, PFAM. Then databases used for next generation sequencing, that is called a sequence read archive. Then database is used in network analysis, metabolic pathway databases, example KEGG, K-E-G-G, then biopsych. Then interaction analysis databases, functional networks. Then databases used in design of synthetic genetic circuits, example XenoCAD. These are all some of the databases. And we can elaborately see about some of the databases. Now we will go to this primary database, one of the primary database, that is called protein databases. So this protein database is called Protein Data Bank, PDB, Protein Data Bank. So this PDB, it is a primary protein structure database. It is a crystallographic database which is used for the three-dimensional structure of a large biological molecule like protein. Okay, protein is a biomolecule. So the name itself says that it is protein data bank, PDB. So this PDB achieve the three-dimensional, that is 3D structures of not only proteins but also all the types of biologically important biomolecules. For example, nucleic acid fragments, RNA molecules, large peptides, example antibiotic gramicidine, and complexes of protein and nucleic acids. So the database which holds data that is derived from mainly three important sources. One is from structure of any protein determined by X-ray crystallography method, and structure analyzed by NMR experiments. NMR means nuclear magnetic resonance experiment, and structure derived by molecular modeling.
According to a report of 2014 Molecular Biology Database Collection in the journal Nucleic Acids Research, there are a sum of 1,552 databases that are publicly accessible online. This does not include online services without publication in peer-reviewed journal or being developed by commercial companies. Thus, the actual number of online databases could well exceed this number. Considering the continuously proliferating number of biological databases, it becomes increasingly daunting and time-consuming to navigate in the huge volume of databases of interest.
And this is the main home page, web page of PDB, Protein Data Bank. Anyone want to open, then they can go to the Google and they can open the home page and they can see the recent home page of the PDB. And this is actually the home page of PDB 2 years back, June 2020. At the time, you see that SARS coronavirus was very popular, so they have taken the molecule of the month, SARS-CoV-2 spike protein. It is the spike protein of the coronavirus. This spike protein actually binds to receptors on the cell surfaces of our body, and it is a target for vaccine development. 2020, at that time there was no vaccine. Now it has been developed. Then primary protein database, another example is PIR. What is PIR? Protein Information Resource, PIR, or it is otherwise called PIR PSD. That is Protein Information Resource Protein Sequence Database. So this PIR PSD, it is a collaborative work between the PIR and the MIPs. MIPS. What is MIPS? Munich Information Center for Protein Sequences, which is located at Germany. And another one is JIPID. It is nothing but a Japan International Protein Information Database located at Japan. So this PIR PSD, it is now a very comprehensive, non-redundant, expertly annotated object relational database management system, that is called a DBMS. It's a very important database management system database. So it is a unique characteristic of the PIR PSD. It is its classification of protein sequences. It is based on the superfamily concept, protein superfamily concept. So the sequence in this PIR PSD is also classified. It is based on homology domain, that is homology domain of the protein and the sequence motif of the proteins. So this PIR is giving almost all the information about primary structure of the protein. That is why it is called a primary database. And this is the PIR, Protein Information Resource home page, web page. And you see that here you see that iPro link is there, PubMed connection is there, all these things are there, text mining tools are mentioned here. And you see that if you want to see the latest web page, you can go to the Google and you can open it. And this is again the home page of PIR. Then we will come to another primary database, but this time it is not protein, it is nucleic acid. So primary nucleic acid databases. Nucleic acid databases, in that we have DNA databases, because DNA is one of the nucleic acid. So primary databases, under that we are going to see DNA databases. So the International Nucleotide Sequence Database, that is called INSD, International Nucleotide Sequence Database, which consists of the following databases under this category. One is called DNA Data Bank of Japan, that is DDBJ, which is actually controlled by National Institute of Genetics, NIG. Then another one is called EMBL, European Bioinformatics Institute, EMBL. It is controlled by European Bioinformatics Institute, that's European Molecular Biology Laboratory. Then another one is called GenBank. It is controlled by National Center for Biotechnology Information. We'll see about DDBJ first. What is DDBJ? DNA Data Bank of Japan is called DDBJ, which is located at Japan. Another one is called GenBank, controlled by USA. And third one is European Nucleotide Archive, which is located at Europe. All these three are repositories for nucleotide sequence databases, that is DNA and RNA sequences. So these nucleotide sequence data from all organisms are stored here. All three except nucleotide sequence submissions, and then they will exchange new and updated data information on a daily basis to achieve optimal synchronization between them, because all the three are functioning put together, combined way. These three databases, they are called primary databases. They house original sequences as data, that is experimental sequences as data. That's why they are called primary databases. All these three collaborate with Sequence Read Archive, SRA, which achieves raw reads from high throughput sequencing instruments. So that is why they are all primary databases. Let us see secondary databases of nucleic acid databases. Example, 23 and ME's database, HapMap, then OMIM, that is Online Mendelian Inheritance in Man, which talks about inherited diseases, then RefSeq. These are all some of the secondary nucleic acid databases. This is the DDBJ data, DNA Data Bank of Japan homepage, web page of the DDBJ. Bioinformation and DDBJ Center, and news from Bioinformation and DDBJ Center is there. This is the homepage.
Having highly different functions, almost all biological databases shared similar architectural. Each consists of 3 tiers of software. At the bottom is a database management system, or DBMS, that manages a collection of facts. At the top is the web browser that transmits requests for data to the database and renders the responses as web pages. In the middle is a software layer that mediates between the DBMS and the web browser to turn data requests into database queries, and to transform the query responses into hypertext markup language, or HTML. In addition to the gene database and PDB, there are many more biological databases designed for different purposes. The table here shows some of the most commonly used databases in bioinformatics that might help you in your research.
Dear viewers, let's go for a small quiz now. Which of the following is a primary database for nucleotide sequences? UniProt or PDB, that is Protein Data Bank, or GenBank or KEGG, that is KEGG. Give your answer now. Next one. What type of information does the Protein Data Bank, that is PDB, primarily store? Nucleotide sequences, or protein sequences, or three-dimensional structures of proteins and nucleic acids, or metabolic pathways? Friends, the answer is three-dimensional structures of proteins and nucleic acids. Friends, next one is, which database is known for storing protein sequence and functional information? GenBank or EMBL-EBI or UniProt or DDBJ? Give your answer now. Which database is specifically designed for the retrieval of biomedical literature citations and abstracts? GenBank or PubMed or SwissProt or ENS EMPL? Give your answer. Next one, KEGG. It is a database used primarily for what type of biological information? Genetic variations or protein sequences or metabolic pathways and molecular interactions or nucleotide sequences? What is your answer? Tell me now. Friends, the answer is, see on the screen.